So I would like to thank the organizing committee for giving me this opportunity. My name is Lee. I will present today the work I did in my first postdoc in the Shaashua lab in the Weizmann Institute, in which we studied how BRCA mutations in the cancer cells shape the stromal microenvironment of pancreatic cancer. Pancreatic cancer is a very aggressive cancer type with a very poor survival rate of 11%. Several somatic mutations are known to underlie this disease, such as KRAS and P53. However, it was also shown that BRCA mutations elevate the risk of developing pancreatic cancer in 2 to 4 folds, and these mutations are evident in 5 to 15% of the patients. In pancreatic cancer, it's very interesting that up to 90% of the cells are actually stromal cells and not the cancer cells themselves. And this is why many groups are studying these cells. So cancer-associated fibroblasts, and in short, CAFs, are the major cell type in these tumors. They are now known to be heterogeneous, and this is very important because if we want to target these cells that are known to support the disease in all of the stages, we want to know which CAF subtypes we have, what are the functions, and which functions we should inhibit. It has been shown that although CAF are genomically stable, they are transcriptionally rewired by external cues, and it is not known up until now whether germinal mutations in the cancer cells may contribute to this heterogeneity of CAFs. So we hypothesized that the stroma of BRCA mutant patients will be different than the stroma of BRCA wild-type patients. To that end, we assembled a cohort of 42 PDAC patients in collaboration with David Kelson from MSK-CC and some other clinicians. And out of these 42 patients, 15 had BRCA mutations. We started by multiplex immunofluorescence, trying to understand which CAF subtypes do we have there. So we excluded all the CD45 and cytokeratin cells, and we found 3 CAF markers, which are mutually exclusive: alpha SMA, clustrin, and HLA-DR, which were in general showing most of the stroma. We then compared BRCA mutant wild-type patients, and we found that clustrin is elevated in the BRCA-mut stroma, and this is quantified here. We then wanted to know whether there's a difference in the transcriptional signature of these different stromas. And for that, we used fresh frozen samples from patients, BRCA mutant and wild type. And by laser capture microdissection, we dissected stromal-rich regions followed by RNA sequencing. We found that there's indeed different transcriptional signatures in these patients. And to understand which protein may underlie all these effects that I've shown you of the different CAF composition and this heat map, we used the IPA software, which suggested HSP90 as an upstream regulator of all these changes. So HSP90 is a heat shock protein, and that means it's regulated by HSF1, the master regulator of the heat shock response. And very interestingly, clustrin, which I just showed you as a CAF marker that is upregulated in BRCA mutant patients, is also a chaperone that is regulated by HSF1. So we thought that maybe HSF1 orchestrates all these changes in the stroma of BRCA mutant patients. We stained our patient samples for HSF1 when it's located in the nuclei. It's a proxy for its activation, and we found that within the stromal cells, there is a higher activation of HSF1 in the BRCA mutant patients. And then we wanted to go to more functional assays. For that, we used the KPC organoids model, which is a very common pancreatic cancer model, and used it also for in vivo studies and to isolate CAFs. I will explain a bit more in a second. So first, we wanted to see whether HSF1 regulates clustrin in our system. For that, we used pancreatic stellate cells, which we isolated from naive mice. Pancreatic stellate cells are the naive fibroblasts of the pancreas. We exposed these fibroblasts to a condition medium from KPC organoids, so for condition medium from the cancer cells. And then using anti-HSF1 antibodies, we checked whether there is a binding to the clustering genes. And we indeed found that HSF1 directly regulates clustering in PSCs following exposure to cancer conditioned medium. We then wanted to know whether there's a difference in after exposure to a conditioned medium of BRCA mutant or wild type cells. For that, we transduced the KPC organoids with SH for BRCA2 or non-targeting control. And then we exposed the same naive fibroblasts to either KPC conditioned medium of the targeting control or a knockdown of BRCA2. We found that both cancer conditioned medium exposures elevated clustering, but the BRCA2 knockdown elevated much more. And this was completely blocked when HSF1 inhibitor was added, suggesting that the clustering expression in these PSCs is dependent on both BRCA mutations in the cancer cells and the activation of HSF1 in the PSCs themselves. We then wanted to know what's the function of these CAFs, and I'm not getting into all of the data. You can find it in the bioRxiv version, but the transition we found from SMA positive CAFs to clustering positive CAFs suggests that they have an immune regulatory effect. So we started on the left here by checking CD8 positive T cell activation. For that, we isolated CAFs from tumors that we injected. So those two cell lines or organoids lines that we transduced, we injected them into mice and then isolated both types of CAFs from both types of tumors. And we exposed T cells to these CAFs in the same well, while activating the T cells. And we found that when T cells are exposed to CAFs from BRCA knockdown tumors, they are less activated, suggesting that indeed there is an inhibition of this activation of T cells. To understand which ligands on the CAFs may do this effect, we used ICELNet, which is a computational tool that gives a score of interaction of ligand and receptors. So we used the list of immune modulatory ligands on the CAFs and compare that to the list of T cell receptors on T cell receptors, and we found that 6 out of these 7 signaling axes were indeed higher in the BRCA2 CAFs, suggesting that CAFs from BRCA2 knockdown tumors has higher immune modulatory effects. To validate that, we used flow cytometry, and we found that both PVR and Nectin-2, which are the modulatory ligands on the CAFs, are indeed higher in these CAFs. So in summary, we found that in BRCA mutant cancer cells, they shape the stromal microenvironment in a different way. They secrete some factors that causes the activation of HSF1 in the PSCs, the naive fibroblasts. That causes a shift of CAF composition towards more clustering positive CAFs, which has an immune regulatory effect. I didn't show all the work, but this is what I chose to show now. And I would like to thank all of the ScienceAbroad lab, both Ruti for mentoring and all my friends in this lab that helped me a lot, and for all the collaborators without which we couldn't do this work too, and the funding agencies, of course. And thank you for listening, and I'm open for questions.